All right. So looking at the syllabus here, we are about a week behind as far as like the stuff I'm lecturing about. I'm not too worried about it because this stuff over here on the last kind of 2 weeks, well, last week and a half of lecture, not a whole bunch to talk about. So the stuff that I'll be able to kind of, there's already room for flexibility kind of built into this last week and a half, the semester. So I'm not too worried about getting through with everything lecture wise in the course. Um, as far as quizzes, I've been saying that the quizzes are now going to be due later than they are listed in the syllabus. From every quiz from now on, I'm sending an email to the entire class on Blackboard stating the quiz is posted and giving you the due date. Usually the due date is, at least 3, maybe more like 5 days after I send the email. I think it's usually more like 5. And I did that recently for the heart review quiz and the ECG quiz right here. Tonight, I will post the blood pressure quiz. And then after that, there'll be 3 more quizzes left, and that's it. And really, yeah, after, let's see, today is the 11th. So really, we only have after we have basically today and 4 total days of lecture. And then we have basically this week and a half break. And I'll point out, your first draft of project 2 is going to be due here. So this is where we are now. So that's 1, 2, 3, and I'm going to make it towards the end of this week. So this will probably do like Friday of that last week. So we basically have about 3 weeks to do everything for project 2. So there are some groups now that are already done coding. I've actually looked at some people's coding, and there are some groups that are done. I'm hoping everyone has at least started the coding. And I will reiterate that I'm more than happy to help you to talk to you, counsel you regarding your coding. I will still offer an extra credit for groups that will meet with me tomorrow to talk about their coding. I will not offer 3 points like I did last week, but I'll still offer 2 points for people. If you've already met with me, then you probably already maxed out on your 3 points. But for groups that have still not met with me to talk about their coding, I'll still do it tomorrow during the normal lab time, Friday rather, to talk about during the lab sessions to talk about your coding. And I'll give you up to 2 extra credit points if your first appointment with me is this coming Friday. So that offer is still on the table, though because it's a week later now, it's only going to be worth up to 2 points instead of 3 points for the people that were on top of things last week. So yeah, all the data files need to be collected it'll probably be around Friday, maybe Saturday of week 15. That's when by then you need to have all 30 or 40 of your participants collected, the data, and then you'll want your first draft of the whole paper due. So it's just over 3 weeks basically to finish project 2. So I'm really hoping the hardest part, as I've mentioned before, I believe is probably going to be the coding part. As far as collecting data, I've already mentioned you can use friends and family for that entire thing. As far as I know, that's the direction everyone plans to go. It's just using friends and family. I'm not too worried about data collection this time. I think the thing will be the biggest struggle for most groups will be the coding inside toolkit. But then also, don't forget, you'll have to write up the whole paper in APA format, and that will be due then. So just over 3 weeks to finish that. So yeah, definitely feel free to contact me to set up a meeting this Friday or some other time. Really, I met with the group last week on Saturday, and I'm definitely okay with doing that as well, of meeting over the weekend. Otherwise, let's see, all to talk about then for the rest of the day is this ERP stuff. I'm continually adding on to the slides, which are already posted on Blackboard. It says EEG and then ERPs in the title. I think the name of the file is, EEG1, EEG2, and then ERPs. Instead of listing all the different ERPs and all the different ERP studies, I'm just saying ERPs in the name of the title for now, towards the bottom of the slides folder on Blackboard. And so I just keep on adding slides to this. And then basically at the end of this unit, there'll just be one long slideshow that contains most of the information about the EEG and ERP unit. So today, we'll start talking about these long latency event-related potentials. Last time we've talked about brainstem potentials have a really short latency. sensory ERPs, which have a relatively longer latency. You know, the P1 and N1 are considered the sensory ERPs. It's estimated they have a latency of like 60 to 100 milliseconds, where the brainstem potentials is like 2 to 10 milliseconds. So super short, a little bit longer. The mismatch negativity has a latency of around 150 to 250 milliseconds. The ERAN has a similar latency as the mismatch negativity, but it's the ERAN, the early right anterior negativity, is studied primarily with respect to musical stimuli. Okay, so we will finish up talking about ERAN. I believe that's where we left off last time. Then we will talk about these longer latency ERPs. So they're longer latency because the latency is usually at least 300 milliseconds for each of these, P300, N400, and P600. So we'll start out talking about the ERAN. Which is right here. So I'll just review a little bit right here. So these are the behavioral findings that we talked about during the first half of the course, the probative task, the harmonic priming task. I went over those last time, so I'm not going to talk about it again. I will remind you, since this is brand new stuff, the ERAN negativity that is larger to a musical event, like a pitch or a chord in a musical sequence, which is unexpected. So the ones that you read about in the ERAN 1 article, there was 2 main chords that were presented, and the researchers measured the neural responses to the 2 chords. An out-of-key chord, which is very unexpected, which technically they called it the Neapolitan chord. And that study elicited a large negativity in this time window of 150 to 250 milliseconds. And then there was the expected chord, the N-key chord, the tonic. This is the chord that music theoretically is supposed to sound most stable and expected to people. And that elicited basically no negativity. ERAN technically is the difference in amplitude responses between these 2 types of chords. And the general trend, the unexpected chord has a relatively bigger negativity than the N-key chord. The ERAN is the difference in amplitude between these 2 types of chords. So I showed you this last time as well. This is the chord by hemisphere interaction from the ERAN article, reminding you that the actual definition of ERAN is the difference in amplitude between the 2 conditions, between the 2 chords, in key versus Neapolitan. Remember, Neapolitan means out of key. And the ERAN is the difference between these 2 chords. It's not just the fact that there's a negativity for this one right here. It's the fact for this is the out of key chord right here. It's the difference in amplitude between those 2 conditions. Technically, that is the ERAN. It's not just this thing by itself. It is this difference between this thing and this thing, the difference in amplitude between the 2 conditions. This is the one in the right hemisphere electrode. And you see a similar pattern in the left hemisphere electrode, FZ frontal in the left hemisphere. You still see a significant difference. You have a negativity and then a relative positivity in the N-key chord. The difference is the ERAN. But one thing to notice, and kind of the reason why this is called the ERAN, the early right anterior negativity, is the idea that the difference in amplitude between these 2 conditions is bigger in the right hemisphere than it is in the left. You get the difference in the right and the left hemisphere. So it's not like all of the processing is occurring in the right hemisphere and none of the processes are occurring in the left. It's occurring in both hemispheres, but the effect is bigger. The difference in amplitudes between these 2 chords is bigger in the right hemisphere than it is in the left. And that is the chord by hemisphere interaction. The 2 chords are N-key and Neapolitan. The 2 hemispheres are left and right. The chord by hemisphere interaction indicates that the difference in amplitude between the 2 chords is larger in the right hemisphere than it is in the left. So that was review. Any questions about this? I know some people had questions about this from the quiz from last week. All right. So here's another interaction. So this, when you have 3 or more factors in a study, then you're going to have many different interaction components in your statistical output. And this study had many factors, and so it had lots of interactions. Not all the interactions were significant, but here's another interaction that was significant: the chord by group interaction. So the chord, once again, is the same 2 chords, the N-key chord and the out of key chord, that is the Neapolitan chord. So those are the two types of chords. But here's a factor that we haven't talked about yet. You read about it, but we haven't talked about it yet, group. And the two different groups in the study were experts, that is people with a lot of musical experience, compared to novices, people with not very much musical experience. And they were comparing in this interaction, they're comparing the size of the ERAN, that is the amplitude difference between the two chords, as a function of these two different groups. Is there a difference in the amplitude, in the size of the ERAN between musical experts and people that are not musical experts? And the chord by group interaction was significant, indicating that the answer is yes. And you can see it pretty clearly on here. One thing I want to point out, and I believe I reiterated this in the notes portion, but if I didn't, I'll make sure to say it. Please notice that there's a factor which is missing from this, that the hemisphere factor, which we had just talked about, that's not part of this thing. And if you look, and that the reason that is because for both the experts and the novices, they showed the basic same pattern, which is both groups showed the ERAN, and for both groups, the ERAN was bigger in the right hemisphere than it was in the left, and you can see it right here. So here are the experts up top, this whole thing, and you can look and you see that, you've seen this before. There's the ERAN, it's big over here in the right hemisphere. You still get an ERAN over here in the left hemisphere, but it's significantly smaller. Similar type of thing in the novices. They have an ERAN in the right hemisphere. And if you look over in the left hemisphere, they have an ERAN, but it's definitely smaller. The difference between these two waves right here in the left hemisphere for the novices is definitely smaller than it is over here in the right hemisphere. So there's no hemisphere interaction. This hemisphere is not part of it because the two groups were actually the same in the sense that for both groups, the amplitude of the ERAN was bigger in the right hemisphere than it was in the left. So that's why hemisphere is not here, because both groups showed the same lateralization of the ERAN, such that it was bigger in the right hemisphere than it was in the left. However, there is this chord by group interaction. And what this indicates is that the amplitude of the ERAN differed between the two groups. And you can tell quite easily by looking at these figures which group had the bigger ERAN. So when you're doing this, really you can look at any of these electrodes, because when you're looking at the chord by group interaction, you don't care about hemisphere. So basically what you're taking is you're taking all of these electrodes for one group and you're smashing them together basically into a single graph, which they don't show, but you have to kind of imagine that. They take all of these different electrodes and basically you kind of average them all together, and then you basically get a single measure of the amplitude differences between the in key and out of key chords for the experts. And then you do the same thing for the novices. You take those different electrodes, you smash them all together, and then you basically get a single graph which shows this is the average amplitude of their out of key chord, the average amplitude of the in key chord. And that's basically what's being compared for the chord by group interaction. And there was a significant interaction. What that indicates, maybe for this one, it might be easiest to just look in the center electrode right here. The amplitude difference for the musicians, their ERAN, it's very large. They have a very large early right interior negativity, very large difference in the amplitudes between the out of key and the in key chords. The novices, they do have a difference between these two chords, no doubt about it. But the amplitude of this difference is smaller in the novices than it is in the musicians. So that's the chord by group interaction, the fact that this difference is really big for the musicians. It's still present for the novices, but it's significantly smaller. This difference is smaller in the novices than it is in the musicians. Questions or comments there? And then here, I believe, is the last thing about the ERAN article. So this was looking at another interaction, the instrument by group interaction. So they had kind of an additional experiment where the thing that was unexpected was not the chord that was played. It was the instrument that was played. So presumably it was, I don't remember the exact details, but it was something like for a while there was a piano playing, you know, some chords, and then all of a sudden the instrument changed from piano to something unexpected like violin, or from piano to guitar. It changed instruments, which is, and so the expectation was built up that, you know, we're listening to a song played on the piano, but then all of a sudden there's this chord that occurs, which was played by a different instrument. And so the thing that was unexpected was the instrument playing the chord. It was no longer the pitches of the chord itself, it was the instrument that was playing the chord. In this experiment, in this part of this paper, that was the thing which is unexpected. But they're looking to see is, you know, is there an instrument by group interaction? So what they did find, first off, is that when the chord is unexpected, there is a negativity in response to that chord. So if I'm playing piano, 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 guitar, when I hear that guitar, my brain notices that something changed. And that is reflected neurally by the fact that there is a negativity that occurs at around 150, 200 milliseconds in response to that unexpected instrument. So in this way, the response to the unexpected instrument is very similar to the response to the unexpected chord, in the sense that both elicit a negativity at around the same time window, around 150, 200 milliseconds. And that's kind of shown here, but these graphs right here are different than the others. And it says down here, I believe, yes, these are difference waves. And so what these are showing is, these are showing the difference in amplitude between the expected instrument, the piano, for instance, versus the unexpected instrument, which would be, you know, like the guitar or the violin or something. And basically any points in each one of these graphs where the amplitude is non-zero indicates that there was a difference in amplitude responses between the unexpected instrument and the expected instrument. And so if you, and then there's experts and novices. And so let's just look right here at this one. You can see right here, there is, this is the negativity right here for both experts and novices. Each one of these, there's this little bump down for each of them. What this reflects is the fact that for each group, whether experts or novices, when you are looking at the amplitude difference between the expected and unexpected instrument, the unexpected instrument elicited a bigger negativity. That's what this is right here. But for this instrument by group interaction, what they're looking at is, was there a difference between the experts and novices in the size of these dips right here? You know, these dips right here, these dips. And you actually look at them, there is a tiny bit of a difference there. You can see the dotted line right there, the solid line right here, the dotted line right here, the solid line. But when they actually did the statistical analysis, although there appears to be differences, those differences were not big enough that they resulted in a significant interaction. And it says it right here. The difference in amplitude between the expected and unexpected instruments did not differ between the two groups. But I do want to mention, there was a significant main effect of instrument, which I did not write here. There was a significant main effect of instrument. And what that indicates is that there was a difference in the amplitude of the responses between the expected and unexpected instrument, such that the unexpected instrument elicited a bigger negativity than the expected instrument. And that occurred, that negativity that occurred for the unexpected instrument, that amplitude was the same for experts and novices, although there's a tiny bit of what looks like a difference there that did not reach statistical significance, which is why the instrument by group interaction was not significant. These are probably the hardest figures to understand in the entire article because they are difference waves. All right. So for the remainder today, we'll be talking about these long latency potentials. We're just moving longer and longer into latency. The ERAN has a latency of like 150, 250 milliseconds. Mismatch negativity is about the same. Now the ones that we're going to be talking about today kind of move into the longer latency potentials. And they have a latency generally of at least 300 milliseconds, although in some studies, the P300 actually has a latency that's around 250. Though on some studies, the P300 actually has a latency which is as long as 900 milliseconds. That's a big range of latencies that can actually occur for this one. But in general, all of these ERPs that we're talking about today have relatively longer latencies than any of the ERPs that we talked about previously. So we'll go over these Each one, I guess we'll start talking about the P300, and I might come back to this slide in a second. So the P300, as its name implies, it's a positivity with a latency around 300 milliseconds. And it's oftentimes studied with respect to oddball stimuli. So you'll be thinking, well, isn't that what the mismatch negativity does too? Mismatch negativity is the neural response to an oddball stimulus, and the answer is yes. The primary difference between the P300 compared to the mismatch negativity, other than the fact that the mismatch negativity is a negativity and the mismatch negativity has an earlier latency than the P300, the primary difference between the mismatch negativity and the P300 is that the P300 only can be elicited if the participant is paying attention to the task. You might recall that I mentioned for the mismatch negativity that oftentimes people will be listening to sounds, like ba, 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 da, ba, 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 ba in their ears, but they'll be actually paying attention to something completely else. They'll be watching a silent video is usually what participants are doing in the mismatch negativity study. And you still get those neural response, the mismatch negativity, even when the participant is not paying attention to the sounds at all. The mismatch negativity, you get that negativity in response to the oddball stimulus, even if the participant is not paying attention at all. The P300, on the other hand, it's a positivity that occurs in response to an oddball stimuli with a latency around 300 milliseconds, but you only get it when the participant is engaged with the stimuli themselves. The participant has to be evaluating the stimuli or paying attention to them in some way in order to get this P300 response to the oddball stimulus. So to say it kind of concisely, the mismatch negativity is pre-attentive. It does not require attention, whereas the P300 does require attention in order to elicit a response. So let's kind of zoom in here to this study. This is a pretty famous study on the P300 published in Science, though it was published in Science in 1965. So this is basically the first, or at least one of the very first studies about the P300 event-related potential. Let's see. So yeah, here's just some general information about the P300. I already kind of mentioned this. The latency kind of varies between 250 and 900 milliseconds. This name P300 comes from the fact that a lot of the original studies, including the one that I'm going to show you right here, the latency of the positivity, as you can see right here, was 300 milliseconds. Since these original studies, though, they've kind of changed different things about the experiments. They might use different participants. And what they find is that the latency of the positivity can actually be a little bit earlier than 300, like 250, or it can be actually way later. If the task is really difficult, then it actually might have a delay of as much as like 900 milliseconds. So the name is a little bit misleading. It comes from the fact that the original studies found that the positivity had a latency of 300. But really the latency can vary quite a bit depending on the participants that you're using and the task that you have those participants performing. But I did kind of mention this. When the task is hard, the latency tends to be longer. When the task is easier, the latency tends to be smaller. Individuals that have cognitive impairment oftentimes will have longer latencies as well. So generally, like you can kind of think of it, the harder the task, the longer it's going to take your brain to process the task, the longer the latency of the P300. Similarly, if there are individuals that have some type of cognitive impairment, all tasks will be a little bit harder. Therefore, the latency of the P300 will tend to be longer for people with cognitive impairment. And so the P300 it's an amplitude peak. It's a positivity that occurs somewhere in this time window, usually around 300 milliseconds, but sometimes a little bit after. And what it's thought oftentimes to reflect is, well, at least in this particular study, this one Sutton study, 1965, is uncertainty or surprise, a surprising event. I mentioned it's oddball paradigm. And so in an oddball paradigm, there's one stimulus that occurs, which is very likely to occur, the standard. But then there's another stimulus which occurs, which is unlikely to occur. It's the oddball. And so the amplitude of the P300, the positivity is thought to reflect to some extent uncertainty, something that occurs when it's not likely or when it's not probable. So this study, they used a cue. This is a study that you has been assigned as a quiz. And so there was a cue, and on each trial, that cue was either something like a visual cue, which was like a light flash, or it was a sound that occurred. And each one of those cues predicted a test stimulus that would occur, I think they said it was like 3 to 5 seconds later. So there was a cue, and it was one of two things. And that cue predicted what the next thing would be with some probability. So in like one experiment, or so for instance, yeah, they did different experiments, but like right here, these are data showed, these are the brain responses in response to the test stimulus. And the test stimulus here is a sound. So there's a cue, and that one of the cues predicted the sound would occur 66% of the time. There'd be a cue, and then following whatever that cue was, the sound would occur 66% of the time. Then there was another cue that when it occurred 33% of the time after that, the sound would occur. And so in both of these situations, both of these waves right here, they're measuring the brain's response to the sound, but the cue that preceded that sound was different. And the thing that's important about that cue is it predicted with different probabilities how likely that sound would be to occur. So what the researchers think they're measuring is, how does your brain, like both of these waves are in response to the exact same thing, the sound. The only thing supposedly which is different about these is how likely was the sound to occur. In one situation, the 66%, the dark line, the participant had believed with high probability, yeah, the sound is probably going to be the thing that I'm about to hear, and then bam, the sound occurs. And this is the brain's response to that sound when the brain is expecting that sound to occur. And then the dotted line right here, it's still in response to the sound, but here the participant is not quite as confident that the sound is going to occur. There's only a 33% chance that the sound is going to occur here. And this is what the brain looks like when the sound occurs, but the brain was not expecting the sound to occur. And if you look, there's a difference right here. There's actually a difference right here too, but this is the difference that the researchers focused in on. And if you go down here, you can see that this difference is occurring right at 300 milliseconds. So there's no time underneath this graph directly. In order to see the time, you have to go way down to the bottom of the figure. And you can see it's about 300 milliseconds right here. There's this big difference, and this is positive is down in this graph, negative is up. So the difference right here basically shows that the brain's response to the sound has a bigger positivity at 300 milliseconds when the likelihood of that sound occurring was relatively small, 33% chance, compared to when the likelihood of that sound occurring was a little bit higher, 66% of the chance. So a bigger positivity when the sound was unlikely to occur. So this is kind of like the oddball because it's unlikely to occur here. And this is like more like the standard because the sound had a higher chance of occurring here. And then you see kind of a similar thing here, but these are the brain's responses to the light stimulus. So once again, there was a cue. One of the cues predicted the light would occur 66% of the time afterward. Another cue would predict that the light would occur with 33% of the time after the cue. And so these are the brain's responses to the light. Participants are looking at a screen and they'll see some light flashing. And the only difference here, once again, is you're still processing the same light stimulus, but the dark solid line is your brain processing the light stimulus when it's expected versus the dotted line is your brain processing the light stimulus when it's relatively less expected. And once again, the thing they focus, you can actually see there's a difference that occurs a little bit before 300 milliseconds, which is actually slightly problematic. And I think the researchers probably should have paid more attention to this stuff right here. But the researchers in the article, which was published in Science, a pretty famous journal, they were focused once again on kind of the peak amplitude right here. There's the peak amplitude here, and then there's the peak amplitude here. And they were basically saying, well, that the place in the electrical activity where the peak occurs is at 300 milliseconds. And you can see that there's a difference between the two conditions such that the stimulus, which is less likely to occur, the 33% light, has a bigger positivity at 300 milliseconds compared to the same stimulus when it is slightly more likely to occur. So one more thing about the P300, which is not a huge focus on the Sutton paper that you read right here, P300_1. But basically, the P300 basically requires, I mentioned this earlier, the participant to be focusing in, to be processing something about the stimulus. If you have a participant paying attention to something else, then the P300 is not going to be elicited. And in general, the participant has to be kind of consciously engaged with making some type of a decision about the stimulus in order for these P300 differences to emerge. And if you're curious, they do say that in the article that you read, that the specific task that the participants were performing in the Sutton, they're supposed to the answer, they would hear that cue, and in that 3 to 5 seconds in between the cue and the actual stimulus occurring, the participants were supposed to give an answer, or to at least think about what is the target stimulus going to be. Is it going to be a sound, or is it going to be a light? That's what the participants, they were consciously engaged with the task, and they were trying to predict on each trial, will it be a sound, or will it be a light that occurs as the target stimulus? And yeah, as I mentioned, mismatch negativity is elicited even when you're completely ignoring the stimulus, when you're paying attention to something else. The P300 is only elicited, or at least it's significantly stronger, when you are consciously engaged, when you're making some type of decision or judgment about the stimuli. So that's, I'm not going to say, here's an abstract, which I've included. It's from Psychological Bulletin, which is a journal which basically writes, in order to get published, basically you have to find basically every single article, or almost every single article, about a subject, and then you have to write a review article basically summarizing all of the existing literature on a given topic. Here's one. It was published 40 years ago, but the P300 is old. It was, originally developed in like the 60s. So this is basically like 15 years of research on the P300 summarized in a single article. And you can just read that. It kind of says a lot of the same things that I just mentioned. But this is just, these things that I said about the P300. This article basically proves that, there's some truth to many of the claims that I'm making. And a lot of these things are talked to. I'm not going to read this whole thing, but this kind of reiterates a lot of the claims that I've been making about the P300. But I want to point out, this Psychological Bulletin article is a review article, and it's different than this. This is this article that you read that was an empirical study. This is an article that you haven't read. I'm just including the abstract because it's pretty informative. Okay, so now I'm going to talk about the P300 in two different studies. So we just talked about it with the Sutton study from 1965. Now I'll talk about it with respect to the Sternberg task, which if you've taken cognitive psychology, you've almost definitely heard about the Sternberg task. I'll review the Sternberg task anyway. And then also, last time someone asked me a question at the end of class, does anyone ever study these event-related potentials with respect to the Stroop task? And I didn't know the answer, but then I found an article, actually I found a couple articles, but I'm only going to talk about one, which looks at how is P300 influenced in the Stroop task, which is relevant to you guys because you guys know a good deal about the Stroop task since you did it in Group Project 1. So I'll talk about the P300 first and the Sternberg task, then we'll talk about with respect to the Stroop task. So once again, this P300, a positivity at 300 milliseconds after something. And in the Sternberg task, this is kind of like each of these rows of this table represent different potential trials that a person could have or be exposed to on the Sternberg task. So in the Sternberg task, you're given a memory set of items at the beginning of each trial. And your job is to remember them, memory set. So we'll just look at trial 5, because this is most trials would, or trial 5 and 4, these are what most trials will look like, one of these two. There'll be like 4 letters maybe that you have to remember, L, S, C, Y, and they'll present them on a computer screen for like a second. You're supposed to remember them. Then the letters will go away. You'll wait some delay period. And then there'll be a probe item, a single letter, and your task is to indicate, did that probe letter occur in the memory set that you saw a couple seconds ago? So it's a really easy task to get the answer right. So here, BKVJ was the memory set. The probe item was M. M is not one of these letters, therefore the answer is no. For this one though, LSCY, C was part of that. So if the probe item is C, the researcher says, what about C? You would say, yeah, that was one of the letters in the original memory set. So I would say the answer is yes here. So as I said, getting the answer correct in the Sternberg task is easy. It's trivially easy. So the dependent variable in this study is not accuracy, it is reaction time, the amount of time it takes to get this answer correct. to correctly say no or to correctly say yes, this is the dependent variable is reaction time. So this is kind of like a schematic diagram of the Sternberg task. So you have some memory set, and then the researcher provides the probe, and you have to, and it says the timer starts running here. It's measuring reaction time. And then you see that probe letter, and you have to think, was the probe in the original memory set? And there's something going on in your mind. I mentioned this is common in cognitive psychology. Cognitive psychology tries to understand what is going on in people's minds when they're doing different things. And that's exactly what this Sternberg task is trying to measure, is what are people doing when they're performing, comparative task, indicating whether something was a member of a memory set from earlier. So this is the main part right here, which the researchers are interested in studying. There's presumably some type of scan and comparison that occurs where you're looking at the probe item, and then you're having to kind of scan some representation in your memory of the items that were in the memory set, and then you're comparing that probe item to each of the items in the memory set. And the researchers are interested in, how are people doing that? How are people comparing this one thing to the representation in their memories? How is that going on? Then people come up with an answer, yes or no, and then they type a response in a computer. So once again, this is what the researchers were really interested in. What exactly is the nature in the minds of people of this scan and comparison? So this is taken from a cognitive psychology textbook. And it's showing, first off, these are the actual data from the Sternberg task right here. And so a couple things to point out here. The memory set, this is the amount of things in that memory set that you have to remember. So sometimes you'll see a single letter and you have to remember a single letter, which is super easy. But sometimes you'll have like 5 or 6 letters in that memory set. So you'd be like, K, T, R, L, N, P. That would be 6 letters. And then you would wait a little bit, and then you'd see a single probe, and you have to indicate, is that probe, was it in the memory set that you saw earlier? The dependent variable, as promised, is reaction time. The blue circles are those trials in which the answer is yes, the probe was in the memory set. The red circles are trials in which the answer is no, the probe stimulus was not in the memory set. And the main finding, which is kind of cool, but maybe not that surprising, is that as memory set size increases, the amount of time it takes to produce the correct answer increases as well. So when that original set size only contains 3 items, your reaction time to get the correct answer to indicate, was this probe item in the original memory set, you're relatively quick. But when there's 6 items in that original memory set, and you have to compare that one probe item to the 6 items, your reaction time is a little bit longer. So in general, the longer the memory set size, or the bigger the memory set size, the longer a person's reaction time. But it's also important, and I'll explain why here when I go talk about these things over here, it's also important to notice that the yes and the no's basically show the same pattern of responses. The yes and the no's, there's not a significant difference in the reaction times between the blue and the red dots. That is, those trials in which the answer is yes compared to those trials in which the answer is no. And so these things over here, B, C, and D, these were kind of the different hypotheses that the researchers were testing. So these are the actual data. But going in, when they actually wrote their introduction section, they didn't know what the data were going to look like. And they're like, we predict there might be 3 possible things that could occur. And each one of these kind of predicts different ways that participants are performing the task in the mind. Remember, I said we're interested in particular this thing right here, the scan and comparison aspect. The researchers basically hypothesized there's maybe 3 different ways people could be doing the scan and comparison. And these are basically the 3 different predictions. So these aren't real data. These are just like, if this is what people are doing, then the data will look like this. If the people are doing it this way, then the data will look like this. And if people are doing it this way, the data will look like this. So let me tell you what each one of these are. The names, by the way, are down here for each of these. So this first one right here is called parallel search. This one right here is serial self-terminating search. And this one is serial exhaustive. So for the parallel search right here, parallel means participants, they get that probe item. And when they're doing the comparison, they're comparing that probe item to all of the items in the memory set simultaneously. And so this makes the prediction, if you're comparing that one thing to all of the things in the memory set simultaneously, then this makes the prediction that even as memory set size increases, there'll be flat reaction time profiles. You're comparing this one thing to all of the things simultaneously. So it doesn't matter how many things there are. You just do everything at the same time, and then you output a response, yes or no. And so that theory, the simultaneous theory, predicts a flat profile such that the more things there are in the memory set size, it doesn't matter. You're still going to have a flat profile of reaction times. Reaction times will not increase, even if you have a lot of things in the memory set. So that's theory one. This is the serial self-terminating. Make sure I'm getting that right. Yeah, serial self-terminating. And this one has an important difference. This is the only hypothesis where it predicts a difference between the no and the yes trials. And this is an important thing to understand. The difference between why it makes different predictions between the no and yes. So serial self-terminating. Serial means doing something sequentially. So you take that probe and you compare it to the first thing in the memory set. You get the answer, yes or no. If the answer is yes, if the probe matches that very first thing in the memory set, then you terminate, you're done. However, if the answer to that first thing is no, then you go to the next thing in the memory set and you look at that. Is the probe item the same as that second thing? Yes or no? If the answer is yes, you're done. But if the answer is no, then you have to go to the third thing. If the answer is no, you have to go to the fourth thing. And if the answer of that last one is no, then you say, okay, the answer is no. And then if you look though, so the serial self-terminating search, you're going through things sequentially, one by one. And once you know the correct answer, you stop, you output a response. And that predicts a difference between the no and the yes responses. And this is something conceptually that I think is important for you to understand. Why would we predict in serial self-terminating search that the yes responses would be quicker than the no responses? And the answer is that yes responses sometimes require you to only look at one thing. Sometimes that very first thing in the memory set is a match to the probe item, and in which you can output yes, the probe item is in the memory set. So sometimes that very first thing gives you evidence that the answer is yes, but you'll never look at that very first item in the memory set and say, oh, it doesn't match, therefore the answer is no. Because if the answer to that very first thing is no, then you have to go to the second thing, and you have to go to the third thing, and then you have to go to the fourth thing. And you have to go through, when the answer is no, you have to look at all of the different things in the memory set before you can answer no. Me personally, if I were making the hypothesis, this is totally what I would expect things to look like. But if you actually look, these are not what the data actually look like. This hypothesis predicts no responses will on average take longer than yes responses, but that's not what they found. And basically there was no differences between yes and no. So this theory is wrong. This theory was wrong as well. This theory turns out to be the one that is right. This is serial exhaustive, and it's a counterintuitive theory. I never would have predicted that this would be the correct answer, but that's what the data appear to show. So serial exhaustive means that you take the probe item, you compare it serially, one by one, to each thing in the memory set, and you compare it to all of the things in the memory set, no matter what the answer is. So for instance, according to this theory, you take the probe item, you compare it to that very first thing in the memory set. Even if the answer is yes, you continue to look at the remaining items in the memory set. That's what serial exhaustive means. The first answer is yes, but I don't output a yes response until my mind has gone through all of the memory items and made a decision, yes or no, regarding each of those. And so the reason there are no differences between the no's and yes in this theory is because no matter how many things, no matter what, you're going through all of the different things in the memory set size, whether the answer is yes or no. So this theory makes predicts no differences in the reaction times of no and yes. And it predicts that the bigger the memory set size, the longer the reaction time. As you can see, the best match out of all three of these theories, all three of these hypotheses, the one that matches the best the actual data is this one right here, the serial exhaustive. You're going through each item in the memory set one by one. And even if you know that the answer is yes after the very first one, for whatever reason, your mind appears to continue going and comparing all of the things, and then you'll output a yes response. So now that's a lot of explanation behaviorally for this. So I do want to talk a little bit about neurally what's going on with the P300. So I have a couple studies about the P300 here. No, I guess, yeah, it's just one. Yeah, it is a couple different studies on the P300. This first one is comparing what does the P300 look like in the Sternberg task when you have a small set size and a big set size. So they simplified it here. They have small set size condition and a big set size condition, but there's no like really meat in between. They just have small set size and big set size condition. I mean, in this study, they also included two different groups. They had poor readers, that is individuals with this dyslexia, and they were comparing that to typical controls. And they were looking at the P300 latency in response to the probe item. So remember, there's a memory set and then a probe item. Is the probe item in the memory set? And they were measuring the brain's response to that probe item. And so they found a couple things. The P300 amplitude was bigger in the poor readers for bigger set sizes. This is a little bit surprising to some people at least, but this is actually not a completely uncommon thing. So it's a little bit interesting to think of the brain's response being bigger for people that have a cognitive impairment, like people with poor reading. But this is actually particularly common in individuals with reading or different types of cognitive impairments, where their brain actually will respond more during a task compared to a control. And you might be like, well, that's a little bit weird because, you know, they have problems with the task. Why is their brain responding more? You would think that people that have problems with the task, that would mean that the brain is responding less. But it turns out that you can think of it as like their brains are sometimes less efficient. And so as an attempt to compensate for the inefficiency, the brain has to kind of work harder. And so sometimes you'll actually see, yeah, in this study, P300 amplitude was bigger for the poor readers. But the findings that are more interesting, in my opinion, regarding the P300 concern the latency. They found that the P300 latency was longer for poor readers. And they also found that the P300 latency was longest for the biggest set size. And this one I put in bold because this is the thing that I think really gives the most, this really gets to the heart of what the P300 is thought to measure. The P300 is thought to measure some decision that you are making about a stimulus that you're seeing out in the world. And the harder that decision is to make, the longer that decision should take. And we see that in the Sternberg task with the really big set sizes, reaction time is big compared to small set sizes. But when we're talking about the P300 latency, we see the exact same thing, at least in this study. The P300 latency was also longest for the biggest set sizes. So this is cool. We studied reaction time the first half of the semester. Now we're studying some of these event-related potentials. This is a study which kind of finds a link between them. That those trials in the Sternberg task, which produce the longest reaction times, also produce P300 event-related potentials that have the longest latency. So there appears to be a correlation between reaction times and P300 latency. So I actually have the data from this experiment. These are the actual waves. And oops, I'll bring this down a little bit. So I'm not going to spend too much time on this because these waves are a little bit hard to interpret, but you can see the different groups. They had small set size of 3, bigger set size of 5, 3 and 5. Those are the different set size, small and large. And then they have the controls versus the individuals that were poor readers. And you can see, for instance, right here, this is the P300 response. Right here, they have the arrows pointing to it. It's a positivity, so positive is up, negative is down in these graphs. And this is the P300 response. You can see it's particularly big for the poor readers at the biggest set size. But I'm not a huge fan of this graph because it's kind of, so much is going on, it's kind of hard to interpret. So I was appreciative that the authors included this nice, clean, simple table, which kind of summarizes the crucial information about P300 latency, and that's what I'm showing you here. So I don't really care about the difference as much between positive and negative trials. Positive trials are trials in which the answer is yes. Negative trials are trials in which the answer is no. And there's the controls are over here. Those are the normal control participants versus the poor readers. And a couple things to notice that in general, the poor readers P300 latency, which is what is measured here, is longer than the controls. You can see for every comparison, for every condition, whether positive or negative, small or big set size, the poor readers, the latency of their P300 is longer, which, as I said, this makes sense because it's taking them presumably longer to perform the task. So it makes sense that this P300, which is supposedly indexing the actual cognitive performance of this task, it's harder for them, so it should take it longer before the P300 emerges in their brain. The other thing to note is that in general, the bigger set size, 5 compared to 3, results in bigger latencies as well. So set size of 5 compared to 3, 504 is bigger than 464. 5 compared to 3, 606 is bigger than 554. And for the most part, you find that in all these comparisons, there's kind of a little bit of an anomaly there. But otherwise, in every situation, the 5 set size had longer latencies than the 3 set size. I want to get through this. I'll talk about this one next time, but it's basically showing the same thing, but I'll talk about that next class. So I want to now talk about the P300 and the Stroop task. And then I have maybe one more thing. So we're probably not going to get through all the long latency potentials today. So here is the P300 study. Some of the information regarding this citation is shown in the notes notes portion of the slide. So this is, we got to think about what's going on in the Stroop task. There's the compatible condition. So this would be like, you see the word blue and it's written in blue ink. Incompatible would be like you see the word blue and it's written in red ink. They have a condition right here, unrelated. They gave the example, they said this would be like the word town written in red ink. So it's like the word that is actually spelled out is completely not a color. It's unrelated to that. And so I don't actually care about the response accuracy at all, so you can ignore this stuff. All I care about are these two things right here. Response latency just means reaction time for these 3 different conditions. And P300 latency is shown right here. So if you remember, for our Stroop task that we did way back, for group project 1, we found that reaction times tended to be bigger for the incongruent, that is the incompatible conditions, the compatible conditions. So they replicated that right here. That's, good to know. The thing which is actually weird about this is that they actually showed the highest reaction times right here for the unrelated condition. That's pretty weird. I've never seen a study where the reaction times for the unrelated condition were bigger than for the incompatible condition. So there's something weird going on here. It might mean that they made a mistake when they were compiling their data. In fact, I would bet money that they made a mistake because this does not happen. Usually compatible condition is the smallest reaction times, unrelated is in the middle, and then incompatible is the longest. It seems possible to me that they actually flip-flopped these. And that's why I'm calling BS right here. Unrelated is not hard. When you're just looking at if the word town is written in red ink and your job is to indicate the color of the ink, that's easy to do when the word spells out the word town. There's no interference. And so the reaction times shouldn't be big here, and in most studies they're not. So something weird's going on here. So you can kind of ignore the unrelated condition because there's something bogus about that, to be honest. But this stuff right here, this kind of makes some sense right here. The response latency in the Stroop task, reaction times are smaller for the congruent condition than the incongruent condition, just like what we found for group project 1. And then if you look at the P300 latency, so they were measuring the brain's response for each trial of the experiment. You were looking at either a congruent trial or an incongruent trial. And then they were measuring the latency of the P300 response. And the latency is shown here in milliseconds. And the latency of the compatible condition was significantly smaller than the latency of the incompatible condition. And so there's this nice little mapping, or this nice little, I guess you could say it might be a correlation. They didn't technically do a correlational analysis, but it appears that in the Stroop task, those conditions which result in the quickest reaction times will also result in your P300 latency being small. Those conditions that result in the bigger reaction time will result in your P300 latency being relatively longer. So the P300 latency, it's pretty clearly in a whole bunch of different studies connected to performing some type of decision. The harder that decision is to make, the longer the latency of the P300. Okay, so this is the last one I'm going to talk about today because I've been going on for a while now. So I'll talk a little bit about the N400, and this is where we'll start off next time. So the N400 is another event-related potential. It is a negativity with a latency of around 400 milliseconds. And as stated here, it's a response to, the N400 is elicited in response to a word that violates a semantic expectation. So this is taken from a website, and I show you the website in the notes portion of the slides, from some professor who's a researcher. And so they provided some of the data that they have from their N400 experiments. And they actually say what the stimuli are. This is the N400 right here, around right here. Let's see, 100, 200, 300, 400 milliseconds right here. And it's the difference in amplitude between these two waves. Please note, negative in this graph is up, positive is down. This red line is the brain's response when you're hearing a word that is inconsistent or unexpected given the preceding context, compared to this blue line, which is your brain's response when you are expecting something. So let me go back to the thing I showed you at the beginning of class today. I'm going to mute that. All right. Okay, so here, the N400 right here, it is, as I said, greater when the word is semantically unexpected, when it violates your expectation. So an example would be like, if I'm measuring your brain's response right here, leg, arm, knee, knee is very consistent with the expectation because these are all body parts. But if I do it right here, leg, arm, cup, and I'm measuring your brain's response to the word cup, this is going to generate an N400 right here, because this is semantically different than the context that preceded it. It gives you the expectation, leg, arm, that the next thing will be a body part, but it turns out to be something you drink out of. That violates your semantic expectation. Leg, arm, knee, knee does not violate your expectation. So the word cup would generate the N4 of bigger negativity than the word knee. Another example, so this is there's kind of two paradigms used for the N400. There's lists, like here where you just list off words, or you can embed it in a sentential context, meaning you can actually embed it in actual speech. So he planted string beans in his guitar. The word guitar violates your expectation because you don't plant string beans in the guitar. Compared, so you'd have an N400 response here versus he planted string beans in his garden. The word garden is very predictable given the sentential, the context. And so you would not have an N400 here. So going back to this right here, this red line, this is the negativity with a latency of 400. This is the brain's response to the word that is unexpected, compared to the blue line, which is the brain's response to a word which is expected. The N400 technically is the difference in amplitude between these two conditions that occurs in this window, which is around 400 milliseconds after the target word is presented right there. So this is where we'll start next time. And I think we'll be able to finish everything on ERPs next time. But if not, then we will definitely be able to finish everything on the class after that. So call it a day there. And if anyone wants to stick around and chat, that is definitely cool. I'll stop recording now.